Hello everyone, myself Achal Kannake. My topic is Fluorescence Spectrophotometer. The table of content I will cover is introduction, schematic diagram, concepts, light sources used, principle and application of fluorescence spectrophotometer. So here is the introduction of fluorescence spectrophotometer. Fluorescence spectrophotometry is a set of techniques that deal with measurement of fluorescence emitted by substances when exposed to ultraviolet, visible and other electromagnetic radiation. Fluorescence spectrophotometer is also known as fluorometer, spectrofluorometer and photoluminescence spectrometer. It is an analytical instrument used to study the fluorescence property of the sample. It excites the sample with a specific wavelength of light and measures the intensity and spectral distribution of the resulting fluorescence emission. Instrumentation in fluorescence spectrophotometry are light source. The instrument is fitted with light source that can emit radiation in ultraviolet, visible and near infrared wavelength. This source serves as sample's excitation beam. Monochromator. The setup features two monochromator. The first monochromator is the charge of determining the appropriate wavelength for excitation, while the second monochromator is used for accurate analysis of produced light. Detector orientation. The detector, usually a photomultiplier tube, is positioned at a right angle to the excitation beam. This orientation enables the efficient capture of fluorescence generated by the sample in all the directions. Then comes excitation of sample. The sample to be studied is placed in the sample chamber, which is often a cuvette with translucent quartz or glass slides. When the sample is exposed to excitation light, the molecules of the solution become excited and emit fluorescence. Excitation monochromator. The excitation light from the lamp source is routed through an optical system to the excitation monochromator. This monochromator allows for accurate selection and scanning of a specific wavelength range for excitation. Then fluorescence emission. The selected excitation wavelength is subsequently directed into the sample compartment, which houses the fluorescent cuvette. When the excitation light interacts with the sample, it excites the molecule within it, and some of them emit light in response. Emission monochromator. The emission monochromator examines the fluorescence that is emitted in all directions. The monochromator is perpendicular to the entering excitation beam and is responsible for the wavelength analysis. Schematic diagram of fluorescence spectrophotometer. This is the schematic diagram of fluorescence spectrophotometer. Here they have used xenon lamp as a light source. Concepts of fluorescence spectrophotometer. How does a fluorescence spectrophotometer work? The optical layout and key component of the fluorescence spectrophotometer is shown in the figure. The sample being analyzed is placed in the sample chamber. This is the sample chamber and excited with a chosen wavelength of light that is selected by the excitation monochromator. This is excitation monochromator. Fluorescence from the sample is the wavelength selected by the emission monochromator and detected by the photodetector. This is the emission monochromator and this is photodetector. The design of fluorescence spectrophotometer with monochromators for excitation and emission wavelength selection is the most flexible for wide range of applications. The second component of fluorescence spectrophotometer is the excitation monochromator. The excitation monochromator selects the desired excitation wavelength from the white light output of a xenon arc lamp. The white light from the lamp is imaged on the diffraction grating, which has a surface of thousands of finely spaced grooves. This is the diffraction grating, and white light source come from here, xenon lamp. Diffraction from this grooved surface spatially separates the light into its constituent wavelengths. The diffracted light is imaged onto a slate consisting of two metal blades with a gap between them. The slits transmit the desired excitation wavelength and blocks the other wavelength. This is the slits that passes only specific wavelength of light and block the other wavelengths. The center wavelength of the light transmitted by the slit is determined by the angle of the diffraction grating relative to the incoming white light, and the wavelength is chosen by rotating the angle of the grating. The bandwidth transmitted can be adjusted by moving the blades to widen or narrow the gap. The narrower the slit, the more monochromatic the light. Then comes the sample chamber. The sample being measured is placed in the sample chamber of the spectrometer. The most common type is a dye solution in a cuvette. This is a cuvette. The excitation light is partially absorbed by the solution, and the emitted fluorescence is collected at 90° to the excitation light. The advantage of 90° collection is that the transmitted excitation light does not enter the emission monochromator, which reduces the background signal and greatly improves the sensitivity of the measurement. Then comes the emission monochromator. After the sample chamber is in the emission monochromator, which selects a fluorescence emission wavelength that reaches the photodetector and operates identically to the excitation monochromator. Widening the slits in the emission monochromator increases the intensity of the fluorescence and improves the sensitivity, but decreases the resolution of acquired fluorescence emission spectra. Then comes the photodetector. The final component is the photodetector, which converts the fluorescence photons into the electrical signal. The most popular photodetector for fluorescence spectroscopy is the photomultiplier tube, due to its excellent light sensitivity. The PMT is a vacuum tube containing a photocathode, a series of electron multiplier called diodes, and an anode. A high voltage is applied across these components to drive electrons from the photocathode towards the anode. When the photon strikes the photocathode, a photoelectron beam is emitted into the vacuum and accelerated by the applied voltage onto the first diode, where it is multiplied by secondary electron emission. This process of electron multiplication is repeated at each successive diode in the chain, and is why PMT are so sensitive. The multiplied secondary electrons are then collected by anode and recorded. This is a fluorescence spectrum uh, plot against intensity versus wavelength. The this blue color spectrum is the excitation spectrum, and the pink color spectrum is the emission spectrum. Steady state fluorescence spectra are when molecules excited by a constant source of light emit fluorescence, and the emitted photons or intensity are detected as a function of wavelength. The fluorescence emission spectrum is when the excitation wavelength is fixed and the emission wavelength is scanned to get a plot of intensity versus emission wavelength. Now we will see what are the light sources used in fluorescence spectrophotometry. Light sources used are LEDs and xenon lamp. Wavelength emitted by LEDs. Each single LED emits a narrow wavelength range. Multiple LEDs cover a wide spectrum from UV to IR. It provides high intensity. The lifetime is 10,000 hours. Decay in brightness across lifetime, no. Xenon lamps wavelength emitted is 200 to 1800 nanometer. UV and visible light range emitted from one bulb. High intensity. lifetime 400 to 600 hours. Decay in brightness across lifetime, yes, it decays decayed in brightness across lifetime. Now we will see the principles of fluorescence spectrophotometer. A fluorescence spectrophotometer is a scientific method used to investigate molecular and atomic interaction by examining the fluorescence emitted from the sample. This technique operates on the basic principle of radiative emission, which occurs when a molecule absorbs energy at a particular wavelength, displaying a transition dipole moment. When the molecule absorbs this energy, its photon move to an excited singlet state. This photon subsequently undergo a relaxation process, returning to the lowest vibrational energy level of the excited state. During this transition back to the molecule ground state, photons are emitted, resulting in the production of fluorescence. Fluorescent molecules exhibit three primary non-radiative relaxation mechanisms, where the excitation energy does not transform into photon: internal conversion, external conversion, and intersystem crossing. Internal conversion occurs when there is a minimal energy gap between two electronic states, causing electron to shift from a higher to a lower energy state, with the energy being absorbed by the vibrational modes of the electronic state. External conversion leads to energy loss as the fluorophore interact with surrounding solid molecules. Intersystem crossing occurs when the energy level of the excited singlet and triplet state overlap, promoting electrons to transition between these states. Here is a small video that shows how the
Fluorophores, or
fluorescent
fluorescent
molecule is being
molecules, can be
excited.
excited by the absorption of light, causing the molecule to reach a higher energy state. This high energy state is unstable and cannot be sustained, which causes the molecule to drop to a more stable, lower energy state. Then the fluorophore will emit light, causing it to return to its original starting energy state, or the ground state. The emitted light will be a different color from the light that was originally absorbed. Fluorescence is defined as the emission of light through this process of absorbing light. In a sample that has been
Fluorescence is a phenomena that results from a complex energy level that are present in all molecules. A molecule's electron is raised from its ground state S0, which is its initial state, that one of the several possible excited singlet states S1, S2, and so on, when it takes in a photon. The electron then moves to S1, the lowest vibrational level of the main excited state. After this excitation, this decrease is accompanied by the energy releasing mechanism known as internal conversion. After that, the excited state is further relaxed to its lowest vibrational level. After excitation, the S1 state, which lasts for about 10 to the power 9 seconds, the molecule can take several different routes back to its ground state S0. Fluorescence is a radioactive process in which photons are released as a result of energy loss during internal conversion. Interestingly, the energy of this released photon is lower than that of the original one that are absorbed. Advantages of fluorescence spectrophotometer is high sensitivity, non-destructive, and cost-effective. Disadvantages are quenching, photobleaching, limited to certain molecular species. Applications of a fluorescence spectrophotometer is in bioscience. One of the most common application of fluorescence spectrophotometer in bioscience is high precision DNA and RNA measurement. An extrinsic fluorophore, usually ethidium bromide, is added to a DNA sample, which is then fed into a fluorescence spectrometer to determine its concentration. Another current application is single molecule real-time DNA sequencing. Its ability to manufacture long-read single molecule with great precision is projected to be essential to the next genetic diagnostic revolution. Second application is in biochemistry and molecular biology. A fluorescence spectrophotometer is critical for understanding the structure, function, and interaction of biomolecules such as protein, nucleic acid, and lipids. Third application is in pharmaceutics. Thanks. If you have any questions, you can comment down. I will try to solve your queries. Thank you.